Dear learners, today we will be discussing on microbial gene transfer, which is a topic of importance in the field of microbial genetics and in development of recombinant DNA technology. So let's get started with the introduction part. What is gene transfer? It is defined simply as a technique to efficiently and stably introduce foreign genes into the genome of the target cells. The foreign genes is the main protocol of gene transfer, where you are inserting the foreign gene into the genome of the target cells. The direct desirable gene transfer from one organism to another and the subsequent stable integration and expression of the foreign gene into the genome is referred to as genetic transformation. Stable transformation occurs when the DNA is integrated into host genome and is inherited in subsequent generations. The transfer gene is known as the transgene, and the organism that develop after a successful gene transfer is known as transgenic. So we will be studying the three processes in detail. Those are transformation, conjugation and transduction. Let's start with the process of conjugation. The definition says that it is a process of genes transfer via the sex pili between the cells of opposite mating types that are in physical contact, that is, cell to cell contact with each other. History says that for the first time, Joshua Lederberg and Edward Tatum in 1946 presented the evidence for the bacterial conjugation, that is, a process of transfer of genetic material by cell to cell contact. So that particular process is named as conjugation. Okay, and the evidence was first presented in the year 1946. The methodology says that it requires the presence of a special plasmid called F plasmid. Now please listen carefully here that the bacteria have a F plasmid which is referred to as F plus or male. F plus means the donor one. Okay, those that do not have an F plasmid are F minus, F negative or female. F positive stands for male, that is the donor, and F negative stands for the female. The F plasmid consists of 25 genes that mostly code for production of sex pili. A conjugation event occurs when the male cell extends his sex pili and one attaches to the female. This attached pilus is a temporary cytoplasmic bridge through which a replicating F plasmid is transferred from the male to the female. When transfer is complete, the result is two male cells. When the F plus plasmid is integrated with the bacterial chromosome, the cell is called an HFR cell, that is called as high frequency of recombination cell. Here you can see that first of all, this plasmid, that is the donor one, this is a bacterial chromosome and this is the plasmid, that is the donor one, because that is F positive male, is extending its sex pili or the pilus to the F minus cell because it does not contain any plasmid. You can see there is only the chromosome but no F plasmid. This pilus retracts. This is the F positive cell, this is the F negative cell. A conjugation is happening here at this point. The cells pair stabilized and F plasmid nicked in one strand. Transfer of one strand of F positive cells to F negative cell. F plasmid simultaneously replicated in F plus cells. Now see, you can see the replication is occurring here. Now the replication has increased. Synthesis of the complementary strand begins in the recipient cell. After the completion of DNA transfer and synthesis, the cells separate. Now you can see the F negative cell has been transformed to F positive. Both of them contains the F plasmid. This is another method of conjugation in which you can see the F positive cell is there and at this point the conjugation is occurring. This is the point. Okay, recombination between F factor and the chromosome occurs at a specific site on each. Which site? This site. This is the specific site. Insertion of the F factor into the chromosome results. What has happened? You see, this F factor, which was yellow in color, has resulted into an integrated F factor, means it has included itself into the plasmid as an integrated F factor, and this is known as HFR cell. The full form is high frequency of recombination cell. Next we are going to study about transformation. The definition says that it is referred to the transfer of relatively small segment of naked DNA from donor cell male to competent recipient female cell. History says that it was first described by Frederick Griffith in the year 1928. He performed an experiment on laboratory mice and two types of Diplococcus pneumoniae organism which cause pneumonia. He established that one type has rough, that is capital R, non-capsulated cells, and the another one had smooth, that is capital S, capsulated cells. The R type is non-pathogenic while the S type is pathogenic. Okay. In Griffith's experiment, the R type 2 strain did not cause death to mice, whereas the S strain caused death to mice. The heat kill S strain also showed similar results like that of R strains. However, when heat killed S strain were mixed with the R strain and injected into the mice died. This induction in change of R strain was called as transformation. Avery, MacLeod and McCarty in the year 1994 conducted the experiment and demonstrated that heat killed S strain transformed R strain into virulent form which caused the death of the mice. The transforming ability was not altered by the treatment with enzyme or by RNS, but was completely destroyed by the DNase. This finding showed that the DNA has the ability to carry the hereditary information. Here you can see the diagrammatic representation of the experiment, the treatment, the result and the conclusion we draw from the result. First is control, in which injected living type S bacteria into the mouse. Type S cells are virulent since the mouse died. Next control was injected living type R bacteria into the mouse. Mouse survived. Type R cells are benign. Next, type 3 control, injected heat kill type S type bacteria into the mouse. The mouse survived, that means heat kill type S cells are benign. Type 4, that is injected living type R and heat kill type S bacteria into the mouse. Virulent type S strain in dead mouse blood was found. A substance from the heat kill type S cells transformed living type R cells into the virulent type S cells. Here you can see that the type 2 strain in the living non-virulent, it was injected into the mice. The mice survives, no bacteria was recovered. The bacteria was then capsulated in a polysaccharide, that is type 3S living virulent. It was injected into the mice and it dies. Type 3S virulent bacteria was recovered from the dead mouse. Then type 3S heat killed non-virulent. This was injected into the mouse and the mouse survives, but no bacteria were recovered. Then type 2 are non-living, non-virulent and type 3S heat kill, that means this one and this one was combined and injected into the mouse and it dies, and type 3S virulent bacteria was recovered, means this one was recovered. Okay. So, this is the explanation of the whole experiment. This particular diagram, they are showing the experiments done by McCarty and group in the year 1994. This says that a mixture of DNA and RNA is taken. It is treated with RNase and only DNA remains. After that, we add the DNA to the R bacteria and this is allowed to grow on a media plate. We see that after some time only S transformants are produced. A mixture of DNA and RNA is taken again and it is treated with the DNase, only RNA remains, and it is added RNA to the R bacteria and this is taken on the growth medium on a plate. There are no S transformants produced. So, this is how they have proved their experiment. Now, we will study about transduction. The definition says that the transfer of DNA from one bacterium to another via bacteriophage is called as transduction. The phenomena of transduction was first discovered by Zinder and Lederberg in the year 1952. He discovered this while searching for sexual conjugation in Salmonella species. What are the different phases of transduction? Transduction happens through either lytic cycle or lysogenic cycle. If the lysogenic cycle is adopted, the phage chromosome is integrated by the covalent bonds into the bacterial chromosome, where it can remain dormant for thousands of generations. If the lysogen is induced by UV light, for example, the phage genome is excised from the bacterial chromosome and initiates the lytic cycle, which culminates in lysis of the cell, and releases of phage particles takes place. The lytic cycle leads to the production of new phage particles, which are released by lysis of the host. Here we are going to demonstrate the lytic cycle and the lysogenic cycle of transduction. To start with the step number 1, the phage attaches to the host cell and injects the DNA. This is the bacterial chromosome and this is the phage. Step 2 shows that the phage DNA circularizes and enters lytic cycle or lysogenic cycle. This is a common step for both of them. From here the steps get separated. In lytic cycle, the step 3 says that new phage DNA and proteins are synthesized and assembled into the virions. At step 4, the cell lysis takes place, releasing the phage virions. So, phage DNA of double-stranded has been produced here. Lysogenic cycle says that at step 3, the phage DNA integrates within the bacterial chromosome by recombination, becoming a prophage. See, this is a prophage. They are getting transferred into a prophage. Then the lysogenic bacterium reproduces normally. The prophage gets replicated. Cell division takes place, and occasionally the prophage may excise from the bacterial chromosome by another recombination event, initializing a lytic cycle. Here you can see that in the lysogenic cycle, what is happening, the phage is attaching, okay, it is integrated as a part of the DNA, then that is replicating, and they are getting differentiated into two different cells. Cell division takes place at this particular step and replication of GNA material. After that material, genetic material is replicated, the cell division takes place and they are converted to two cells. Okay. What is specialized transduction? Some phage transfer can only take place via particular genes to other bacteria. The phase lambda has this property. To understand specialized transduction, we first need to examine the phage lambda life cycle. Lambda has two distinct phases of its life cycle. The lytic phase and the phage infects the cell, make more copies of itself, then lyses the cell to release the new phage. The lysogenic phase of the lambda life cycle starts the same way. The lambda phase binds to the bacterial cell and injects its DNA. Once inside the cell, the lambda DNA circularizes and then incorporates into the bacterial chromosome by the process of crossover. The specialized transduction continues. Once incorporated into the chromosome, the lambda DNA becomes quiescent. Its genes are not expressed and it remains a passive element on the chromosome, being replicated along with the rest of the chromosome. The lambda DNA in this condition is called as prophage. After many generations of the cell, conditions might get harsh. For lambda, bad conditions are signaled when the DNA damage occurs. When the lambda prophage receives the DNA damage signal, it loops out and has a crossover, removing itself from the chromosome. Then the lambda genes become active and it goes into the lytic phase, reproducing itself and then lysing the cell. Lambda can only incorporate into a specific site, that is called as at lambda, that is attachment. ATT comes from the attachment lambda. The gal gene is on one side of the at lambda and the bio gene, that is biotin, is on the other side. Sometimes when lambda come out of the chromosome at the end of the lysogenic phase, it crosses over at the wrong point. When this happens, a piece of E. coli chromosome is incorporated into the lambda phage chromosome. These phage that carry an E. coli gene in addition to the lambda genes are called as specialized transducing phages. They can carry either the gal gene or the bio gene to other E. coli. Here you can see that the prophage exists in the galactose using host. Okay, so this is the gal gene that is from the galactose, and this is the prophage, and this is the bacterial DNA. The galactose positive donor cell works over here, and the phage genome exchanges carrying within the adjacent gal gene from the host. So here the excision takes place. Okay, and phage matures and cell lyses, releasing phage carrying gal gene, that is the galactose gene. Here it is released from the cell lysis. Phage infects a cell that cannot utilize galactose, that is lacking the gal genes. Okay, this does not have the gal gene, so it is infecting that particular, the phage is infecting this, and it is giving out this gal gene over here. Along with the prophage, the bacterial gal gene becomes integrated into the new host DNA. Here it is integrating itself, and the lysogenic cell can now metabolize the galactose. So there you can see the integrated cell. With this, we complete our presentation on the topic of microbial genetics, in which I have covered in detail about conjugation, transformation and transduction. I hope this presentation has helped you a lot to acquire knowledge about the microbial genetics and the process of gene transfer. Thank you for listening and following my videos.